In this video, we will continue our examination of population genetics. Previously, we talked about the Hardy Weinberg distribution and how the frequency of alleles stays similar from generation to generation, assuming that certain criteria are met. Also, the allele frequencies can predict the genotype frequencies as long as that population is in Hardy Weinberg equilibrium. In this video, we will look at cases where populations are not in Hardy Weinberg equilibrium, and we'll talk about random mating, we'll talk about small populations, and populations that are affected by migration, mutation, and unequal survival. First of all, non-random mating. It happens in two main ways, either by inbreeding, which would be mating between genetically related individuals, or assortative mating, which would be mating between phenotypically similar individuals. In either case, both types of non-random mating leads first to an increase in recessive disorders. And you can see an example of that if you look at the populations from Japan, which is an island population that is definitely more inbred than the European population. You see greater levels of certain types of genetic disorders. Over time, non-random mating leads to a reduction in genetic diversity, and this shows itself by a reduced frequency of heterozygotes and more homozygotes. When you inbreed a population systematically, you finally start to lose alleles. So for example, if you have two alleles for one particular gene, over time systematic inbreeding will lead to eventual fixation of an allele, either big A or little a. The extreme of inbreeding is actually found in the inbred mice strains and rat strains that we've talked about that are used for genetic research. So these ones have been systematically inbred for something like 40 generations, and at that point, all of the alleles have been fixed and all of the genomes are pretty much identical. Another exception to Hardy Weinberg equilibrium occurs when populations are very small. So there are a couple of things that can happen here. First of all, populations are small, you get sampling errors from generation to generation. This is an example on the left here of a population of a fixed size of 20. And you start out with two alleles that are equal to each other, A1 and A2. And over time, each generation, you get a little bit of sampling error. And that sampling error is going to be much greater if you have smaller populations. So this is actually 5 simulations of what might happen in a population size of 20 from generation to generation. And what you see is that the allele frequencies do in fact change. They move away from that 0.5. If you have large sampling error, you can actually get fixation of a certain allele. So for example, in this particular model, this population 1, allele A2 got fixed at around generation 18. So this sampling error is called genetic drift. On the right is another figure. This actually looks at the effect of population size on a little bit larger scale, so looking at 100, 200 generations. And what you see here is that over time you get this variation, but the larger the population size, the less likely that you're going to deviate from that starting point of 0.5. In this case, it's a frequency of heterozygotes. So this population size of 1,000, you just get a little bit of genetic drift over long periods of time, 200 generations, whereas if you have a population size of only 16, you tend to get much larger genetic drift over generations. Another effect of small populations can be found in something called the founder effect. So the founder effect is you start out with a bigger population and then a small population breaks off and founds a new population. You would think that since each of those founder populations came from that original larger population, that each one of those would have the same allele frequency as was found in the larger population. But it turns out, again, that when you have very small numbers of individuals, you can get a sampling error, and you can take these individuals that just by chance all of them have that red allele instead of the blue allele, and that population then will go on to found a new population with a different allele frequency than the population where those original individuals came from. Migration, mutation, unequal survival. First of all, let's talk about migration. So in human history, there's been a lot of examples of migration, and a lot of times those individual populations will have different allele frequencies in their two separate populations. What will happen is after only one generation, as long as you have random mating, you'll establish new allele frequencies based on the populations that are joining together, and that population then will return to Hardy Weinberg equilibrium. Mutation is another thing that can affect whether you're in Hardy Weinberg equilibrium. In general, the natural mutation rate is actually very low, and thus the effect of mutation on the Hardy Weinberg equilibrium is very, very small. So most of the time, we're really not going to worry about the effect of mutation. In the short term, it's a really negligible effect. Finally, unequal survival is also very important. So different alleles may confer different abilities for that organism to actually survive. This is actually natural selection. If you have selection of alleles, of certain alleles, that is going to influence the frequency of the alleles in the population. So the most fit alleles tend to contribute their alleles to more of the progeny, whereas the less fit individuals tend to survive and reproduce less. So this is going to change the Hardy Weinberg equilibrium. From generation to generation, you won't have this maintenance of allele frequencies at exactly the same rate. It's important to remember that fitness is a factor of both the genetic makeup of that organism and also of the environment. And I'll just mention a really interesting example of unequal survival. This is differential fitness in different environments. So this example is of the peppered moth, and the peppered moth exists in two forms, either the dark form, which has at least one dominant big C allele, or the light form, which is little c, little c. The peppered moth generally hangs out on trees, and it turns out that in England in the early 1900s there was a lot of soot in the environment, and this was because of industrialization, and a lot of the trees looked very dark just because of that soot in the air. And it turns out that during that period, the dark form of the peppered moth got an advantage, right? Because the dark form could camouflage much better on the dark colored trees. And so that dark form almost completely took over. Before the industrialization era, it was mostly the light form, right? And so the dark form actually went from only 1% to something like 90% in just a period of a couple of decades. It turns out that recently, trees in England looked more like this, look more normal because there is less air pollution, and the light form of the peppered moth has actually come back. So this is an example of unequal survival based on the environment that individual finds itself in. And allele frequencies don't just remain the same, they will change if there is this selection going on. Another example is found with sickle cell anemia in tropical Africa. So sickle cell anemia is actually a really bad disorder. The red blood cells can form this sickle shape, and if you are actually homozygous for the sickle allele, it is generally considered to be fatal. There's certainly medical intervention that can happen, but in tropical Africa, it tends to be that these individuals are very, very badly off. They can't deliver oxygen to their body very well. Yet in tropical Africa, it turns out that the sickle allele is actually at a pretty high frequency. And people wondered why it was so high when it was such a deleterious allele. The reason turns out to be that if you are actually heterozygous for the sickle allele, so if you have one wild type and one mutant allele, those heterozygotes are actually less susceptible to another really bad infection that's found in that part of the world, which is infection with Plasmodium to get malaria. And therefore, this provides this balancing preference for individuals to have one sickle allele. And therefore, because of that selection, this pretty deleterious allele is actually maintained at a pretty high level in the population. The last thing I'd like to talk about with regard to unequal survival is the really extreme example. And so we've talked about some pretty bad genetic disorders in this class, things like Tay-Sachs disease, where children die when they're 5 or 6 years old. And therefore, if you think about it, what happens whenever you get a child with Tay-Sachs disease is that those two alleles are then removed from the population. So every time we get a homozygote, those individuals completely fail to reproduce and pass on their alleles, and thus those alleles are removed from the population. Now, one thing that is a little confusing is to think, well, how is it that so many deleterious genetic disorders can actually be maintained in human populations when we constantly remove those alleles from the population? The answer to that is that at a certain low level of allele frequency, almost all of the alleles are found in heterozygous state. So just as an example, if we think about the beads that we used to demonstrate the Hardy Weinberg equilibria, so if you had a very small allele frequency, so say you had 3 mutant alleles out of a total of 100, so 97 of one color and 3 of the other color. If you were randomly drawing beads out of that tube, the likelihood of getting a homozygote is very small because there are so few of them. And so the majority, almost all of those mutant alleles in the population are found in a heterozygous state. So what will happen with these harmful disease alleles is that they very slowly build up in the population, and this would be due to that very low rate of natural mutation. And mostly at that point, the alleles are recessive, right? So they are in heterozygous condition. Since there's so few of them, almost all of them are found in a heterozygous condition, and so they're not selected against when they are in the heterozygous state. At some point, that allele frequency will get high enough so that you start getting homozygotes appearing. And at that point, you can start selecting against that allele. And so what happens in populations when you have these very deleterious alleles is that we reach a level of balance whereby the elimination of those alleles from the population due to appearance of homozygotes is balanced by the introduction of new mutations. And so over long periods of time, we can remain at this stable level of allele frequency in that population. And when you have this kind of elimination by selection balanced by mutation, this is actually not Hardy Weinberg equilibrium. I just want to make the point that this is a population that is undergoing selection, and it is a balance, but it is not a balance of Hardy Weinberg equilibrium.